Good afternoon, everyone. So good to see you on this sunny day. I'm Tamiko Brown-Nagin, the Dean of the Harvard-Radcliffe Institute, and I'm pleased to welcome you to this year's Kim and Judy Davis Dean's Lecture in the Sciences. Today, we're joined by the pioneering scholar of genetics and human evolutionary biology, David Reich. Before I introduce our speaker, let me take a moment to thank Kim and Judy Davis, whose support makes this lecture series possible. I'm grateful as well to members of the Radcliffe Institute Leadership Society and all our annual donors. Your generosity keeps Radcliffe programming free and open to the public, and we thank you. Recent decades have seen profound advances in genetics and genomic science. Consider the example of the human genome, that is, the entire set of genetic instructions encoded in a person's DNA. It was just 24 years ago that scientists published the first draft sequence of those instructions, illuminating some 90% of the human genome. This milestone came after immense financial investment and over a decade of dedicated work by researchers around the world. Since then, rapid innovations in technology and research methods have fueled fundamental discoveries about the genetic building blocks of living organisms. And scientists and practitioners are using this knowledge to address some of the world's most pressing challenges. In medicine, molecular genetics and genomic science have enabled the diagnosis of thousands of rare genetic conditions and aided the development of new drug treatments and therapies. For instance, genetic research at Harvard Medical School, the Dana-Farber Cancer Institute, and Boston Children's Hospital became the basis for a new FDA-approved therapy that uses gene editing technology to treat sickle cell disease. Other researchers are using genomics to study how individual behavior, the environment, and genes interact to influence one's risk of developing conditions such as heart disease and many cancers. And in the realm of infectious disease, public health practitioners have leveraged whole genome sequencing to more rapidly pinpoint and stop outbreaks of bacterial foodborne illnesses. Of course, this is only a fraction of the myriad ways scientists are using genomic science and genetics to better understand and advance human health. And while such applications may be the first that come to mind for many of us, the potential of genetics extends well beyond medicine and biology. Scholars and engineers in a variety of fields are experimenting with DNA for a wide range of potential applications, from converting carbon dioxide to help tackle climate change, to crafting digital memory materials that might store the vast data of our information age. Fascinating areas of research and innovation lie ahead, offering both great promise as well as some peril, including profound ethical questions that we must address as our knowledge and capabilities continue to grow. Today, we have the chance to learn about another rapidly advancing area of genetic research, ancient DNA sequencing and analysis. In the last decade and a half, ancient DNA has emerged as a novel tool to examine distant history. This is both a remarkable technical feat as well as the opening of countless new potential avenues for scientific inquiry. Indeed, ancient DNA has already repeatedly produced unexpected results that have stirred passionate debate about the complexities of the human past and how we study it. David Reich is one of the foremost scientists using ancient and modern DNA to examine human history and biology. His lab has produced more than half of the ancient human DNA data reported to date, and he and his team have been credited with developing, to quote a fellow geneticist, some of the most sophisticated statistical and bioinformatics techniques available. As we'll hear about today, David's research has produced many consequential findings that have, in the words of this afternoon's discussant, Carl Zimmer, reshaped our understanding of human prehistory. David is a professor of genetics and human evolutionary biology here at Harvard, as well as an investigator of the Hughes Medical Institute. He's the author of numerous publications, including the New York Times bestselling book, Who We Are and How We Got Here, Ancient DNA and the New Science of the Human Past. His many awards and honors include the Fabio Frazzetto, there we are, International Prize for Physical Anthropology and the National Academy of Sciences Award in Molecular Biology. He's also co-recipient with Svante Pääbo of the Dan David Prize in Archaeology and Natural Sciences, the Wiley Prize in Biomedical Sciences, the Darwin Wallace Award, and the Masary Prize. I'm delighted to welcome David to the Institute today and to have this opportunity to learn about his work. Let me also extend a special welcome to Dr. Pääbo and his wife, Dr. Linda Vigilant, who are here today, as well as David's mother, Tova Reich, who's also a former Radcliffe Fellow. Thank you all for joining us. Before I give the floor to David, let me provide a brief overview of today's program. As I mentioned a moment ago, after his remarks, David will be joined in conversation by the award-winning author and journalist, Carl Zimmer. Carl writes the Origins column for the New York Times, and during the COVID-19 pandemic, he contributed to coverage that won the Times the Pulitzer Prize for Public Service in 2021. Carl is the author of 15 books about science, the latest of which is Airborne, The Hidden History of the Life We Breathe. He's a recipient of numerous awards, including the Stephen Jay Gould Prize, the National Academies of Sciences, Engineering, and Medicine Communication Award, and the American Association for the Advancement of Sciences Journalism Award. His podcast series, The Future of Aging, was released in the spring of 2025, and he is a professor adjunct at Yale University. Thank you, Carl, for being here today as well. Following David and Carl's conversation, we'll open the floor to audience questions. Whether you're watching us in person or online, you can submit questions at any time using the Slido link that is provided on the screen behind me and posted in the chat feature of the Zoom webinar. And now please join me in warmly welcoming David Reich to the stage.
Thank you, Tomiko, for the introduction, and it's really a great honor and to everyone who invited me today. So the title of my talk is Ancient DNA as a Window into Human History and Biology. And I want to begin by talking about this new technology, which I've always viewed as kind of a new scientific instrument with the power that a new scientific instrument can sometimes have. So 400 years ago, there was the invention of instruments like microscopes and telescopes that made it possible to peer into worlds that had not been looked at before. And in the same way, ancient DNA is able to peer into the past and to understand how people are related to each other and to people living today. And what it shows is worlds that we hadn't imagined before. It's very surprising. So the measure of a new scientific instrument is, and the importance of a new scientific instrument is when you use it to look at something that has not yet been looked at before, how surprising the results are. So I hope that at the end of this presentation, and maybe after the conversation with Carl and the questions, you'll come away feeling that this is really very powerful as a new way of looking at the past. Ancient DNA begins with a remain. This is an ear bone from a human in our laboratory. And then in a clean room where the goal is to get beneath the surface of the bone and not to contaminate it, we take a small part that's hopefully DNA rich. We take a little bit of powder, maybe the 40th of the weight of a paperclip. We release the DNA in a solution that removes the protein and mineral content, and then we sequence, often with an enrichment step that improves the efficiency of the sequencer. The talk today is not about the deep integration of ancient DNA into biology, archaeology, and environmental sciences, which is super exciting and is finally now happening. It's really being put to the surface of these other fields. That's another talk that should be given and I could try to give, but I'm not talking about that today. This is a talk whose theme is about 3 examples of how qualitatively new data can falsify assumptions and hopefully change our understanding of the world. So the 3 examples are 1, 2, 3. So 1, I'm going to talk about before ancient DNA, we had a fundamentally wrong understanding of how modern humans, people like us, who appear in the skeletal record about 300,000 years ago for the first time in Africa, relate to archaic humans like Neanderthals who lived in Europe and Western Eurasia. So in 2007, Svante Pääbo, who's in the audience today, started getting genome-scale data from Neanderthals. And this was really a very, very exciting development. And I got a lucky break. So I was a 4th-year assistant professor at Harvard Medical School, and I was studying diseases, especially in African Americans, that had differential risk across populations. Svante, because of side work I had been doing, invited me to be part of an international consortium where my role would be to co-analyze the data to learn about modern human population relationships. It was clear to me and to my colleagues, especially Nick Patterson, who's also here, and who worked with me on this, that this was the most important data in the world. I kind of feel this sometimes when reading about gravitational wave data or something like this, that maybe I should start working on that. But it's just such amazing, exciting, like holy data. It's very special. And I think that that was felt by all of the people in our international collaboration. Svante had an amazing group already in Leipzig, but I think he decided to put together an international collaboration to bring together people with a variety of skills from all around the world to work on it. And it was really an extraordinary experience with people really not competing at all, but just trying to do the best work because they were in awe of the data that we had. For this next 7 years, I spent most of my own time analyzing this data. I told myself I'm going to do a second postdoc, really. And what happened, actually was interesting, over the weekend I had lunch with a colleague at Brandeis, and he asked me, who did you do your postdoc with? And he said, I thought it was Svante Pääbo, so I was very happy to hear about that. I actually did my postdoc with Eric Lander at the Broad Institute, and that work has also, that influence has also been very, very important to me. So the background for this work was that geneticists had convinced ourselves, and I had been a minor part of that as a graduate student, that Neanderthals, archaic humans who had lived in Europe and Western Eurasia, and that we had an amazing skeletal record of and amazing archaeology, had not mixed with modern humans in a way that left a trace in people living today. Though overlapping in Europe from 45 to 40,000 years ago, and though overlapping in the Middle East for tens of thousands of years before that. Here's an example of a paper that argued why that seemed to be so. But analyzing the data that was produced by Svante's group, I soon found evidence that they had mixed. And I was really incredulous because of this background coming very much from this tradition in human DNA that this had not happened. We hadn't seen an ounce of it. We hadn't seen a hint of it. And there were all sorts of weirdnesses about the genome, that all of which could look like Neanderthal admixture. If you're screening through the genome for something weird, then any errors in your sequencing, any errors in your processing might look like Neanderthal ancestry. So we kept trying to make these results go away, but we eventually became convinced after compiling multiple lines of evidence. So here's the simplest of these pieces of evidence. So this is a cartoon of a chromosome, like one of your 23 pairs of chromosomes. And if you take a sequence from a French person, a European, or a Nigerian person, look at a difference between them. There's about one such difference every 1,000 DNA letters. There's about 3 billion DNA letters in a genome. And you ask whether a Neanderthal differs and matches the French or the Nigerian. If the Neanderthal is symmetrically related to these two groups, it should match equally often, but we found much more matching statistically to French people than to Nigerian people. And this was true not just for Europeans, but for East Asians. It was also true not just for West Africans, but for Southern Africans and Central African hunter-gatherers. So it was a difference between sub-Saharan Africans and non-Africans that we saw consistently. Another thing we did is we replaced this European with a second Neanderthal we got data from, and the skew became much, much bigger, about 50 times bigger. Because it was 50 times bigger, we could learn that French were 2% of the way to a second Neanderthal, so it was 2% gene flow into Neanderthals. That was the interpretation. And an equally big surprise was that there was as much signal of Neanderthal affinity in East Asians and in Papuans from Melanesia as there was in Europeans, despite that Neanderthals never had been in these places. So that was very surprising to us too, because the obvious place that mixture would have occurred was in Europe. And if you asked physical anthropologists where mixture with Neanderthals happened, if it did happen, some might say it did happen, but they would have argued it was Europe. But we saw there was no evidence of a special signal in Europe. In fact, there might even be less Neanderthal ancestry in Europeans today than in East Asians. So the next thing that we did, among many things, was we built maps of where in each person's genome you can find the Neanderthal ancestry. And it turns out to be much easier than one might expect because it's so distinctive. You move along the genome and there's sharp evidence that there's high rate of matching to Neanderthal, for example, or lineages that are very deeply diverged. So even though these segments are maybe only 40,000 DNA letters long, you can see them sharply and be confident of many of them in terms of their locations. So in 2014, we and others built a map of where in large numbers of people's DNA these Neanderthal segments occurred, and you could pile them up in large numbers of Europeans and East Asians. The average is 2%, but you see there's spikes where it's more than 2%. And there's also, which are possibly evidence of natural selection in non-Africans for segments of DNA derived from Neanderthals that maybe even might have been pre-adapted to the Eurasian environments modern humans were moving into from Africa, maybe cold adapted, for example. There were also deserts, places where Neanderthal ancestry was absent, that are potentially places where in the mixed individuals it was essential to carry a modern human genetic variant because it was particularly useful for the population. There were places in the genome that were systematically depleted in Neanderthal ancestry, especially chromosome X, one of the two sex chromosomes, and sperm-related genes. And this is the characteristic in many species of male hybrid sterility. So if you take two groups that are at the edge of biological incompatibility and mix them, quite rapidly there's kicking out of the mixed genome of DNA around segments that are related to sperm biology in males, and also on the X chromosome, which are associated with male infertility often. So we think this is circumstantial evidence that there is male hybrid sterility associated with this mixture process. We had a huge piece of luck. Svante's group was working with a group of scientists in Russia, and in 2009, I think, approximately, they shared with Svante's group a pinky bone that size from excavated in a cave in Siberia, Denisova cave. And when DNA was obtained from it, it was high-quality DNA, and it was a something else. It was neither Neanderthals, these four here. It was not modern humans. It was a something else. It looked like a distant cousin, but very, very distant, hundreds and hundreds of thousands of years from Neanderthals. So this was extremely interesting because there were no tools found near it. There was not a big skull. It was just floating there, but it was a something else, a sort of remnant of a group of people that had been living, and we didn't know what they looked like, but we had their genome. What we found, we applied the same tools that we had developed for studying mixture with Neanderthals, and immediately we saw that these Denisovans had a high degree of connectedness to one particular group in the world, which are New Guineans. So these are people who live in a very equatorial part of the world, and you see much more matching to New Guineans than to East Asians, for example, or to any other group in the world. And when you do the same analysis, it's not even 2%, it's more like 3 to 5% Denisovan ancestry New Guineans. Now, Siberia is very far from New Guinea, so what you're seeing is remnants of a population that was spread over a vast region, probably much bigger, actually, geographically, than the Neanderthals were spread over. We now have then built maps of where the Denisovan ancestry is present, and if 3 to 5% is what you see in New Guinea and Australia, about a 20th of that is what you see in most of East and South Asia. And there's an especially large amount in different parts of the Pacific who are impacted by Papuan-like ancestry. Just a few months ago, we now have a Denisovan cranial record. Xiaomei Fu and her colleagues obtained DNA and protein sequence from this extremely ancient skull and is matched as Denisovans, so we're beginning to know what they look like. So it's been clear that modern and archaic humans mixed everywhere they met. It's not a rare thing for people to mix with people who are quite different from them. It's in fact the rule. So this is work almost entirely from Svante's lab. Here's a paper I was involved in 2015. This is the Awasa jawbone from a cave in Romania from about 40,000 years ago. This person had a Neanderthal 4 to 6 generations back in their family tree, much more Neanderthal than random people that we had data from. Another study by their group found 3 individuals in Bulgaria that also were like that. In Svante's group, they also found, after screening thousands of tiny, uninformative bone fragments in Denisova cave, most of which were animals, they looked at their protein sequences and saw which of them are human, and of the very small number that were human, they got sequence from one, and it turned out to be a human, and it was a first-generation hybrid of a Denisovan and Neanderthal. It's amazing. So this world was full of people who were a mixture of very different humans. Today, we're very similar to each other. Even the most different people are at most maybe 200,000 years separated with little gene flow. But 70,000 years ago, there were at least 5 groups, far more different from each other than any groups living today. A question that I have is, do we still have a fundamentally wrong model of archaic modern human relationships? This is approximately the picture we now have, without even talking about the recent gene flow between Neanderthals and modern humans in New Guinea, Melanesia, and out of Africa. There's substructure in Africa, there is a deep lineage going into Denisovans, and the standard model has this very interesting event that I hadn't talked about, which is about 5% contribution of African modern humans to Neanderthals about 300,000 years ago. However, the mitochondrial DNA you get from your mother and the Y chromosome you get from your father, these are, in Neanderthals, something that's related within 300,000 or 400,000 years to modern humans. So if you believe this model, it must come from this event. The probability of that happening is 5% times 5%, so a very, very small number. It's been argued that maybe natural selection for the modern human mitochondrial DNA and Y chromosome, which was better adapted, could explain this, but other work has shown that the rest of the genome for modern humans was strongly selected against, so it's a very strange story. So it makes one think, are we missing something important, and is there an alternative configuration about the relationships amongst these lineages that might explain the data? Maybe if we think very hard, we'll find it, or maybe we'll cut some Gordian knot and get more ancient DNA. 2, so before ancient DNA, we had a fundamentally wrong understanding of how human populations migrate and mix with each other. So I'll tell you a little bit about my own story. My own story generating this kind of data started with a frustrating faculty search I was invited to be on as a guest part of the committee here in 2011. So as part of this work in Leipzig that I was intensely involved in, I was really convinced that ancient DNA from the last 10,000 years, a topic that Svante wasn't interested in working on, would be transformative for understanding how the world got to be like it is today. There's people speaking 7,000 languages around the world, and that diversity reflects largely things that have happened in the last 10,000 years. And this technology, which gets DNA out of people 150,000 years ago, maybe it will be really easy for people who lived 3,000 years ago. So let's do that. And I was desperate for someone to be recruited that I could work with. The search committee didn't even invite quite extraordinary applications that we got on the basis that this is, quote, not relevant to human evolution. And it became clear that no specialist would be invited. It fell between the cracks in the university. So I retooled my lab very intensively. And we also optimized a new methodology for ancient DNA enrichment, focusing on industrial scale production of high quality data, which was what I had done my postdoc with working with Eric Lander at what became the Broad Institute. As of a couple of years ago, there were now more than 10,000 individuals as a result of this series of innovations by our group and others. A prevalent assumption until 2015, which was a reaction to the Nazi abuse of archaeology for politics, was that changes in the material culture of record of Europe, which are very dramatic, 5,000 to 4,000 years ago, associated with the spread of certain types of ways of burying people and types of goods and pots, were unlikely to be due to migration. The Nazis had argued that these spreads were associated with the Aryans and were the beginning of the spread of Indo-European languages, and that they spread out out of northeastern Europe and Central Europe and Germany, and that this what defined the area that was the German homeland and was part of building a claim to territory. That was deconstructed in the subsequent decades, and it became very difficult in archaeology to argue that migration had occurred as a result of this. And it became a sort of assumption that when cultures change, it should be assumed to be communication of ideas, quote, pots, not people, that people are communicating with each other. Like today, people use cell phones all around the world, but they have different backgrounds. But it was not possible to test this at all. You just couldn't tell if people were related to each other until the advent of ancient DNA, when you could tell exactly. You can see how people are related to each other. So one of the things that we've been intensely involved in our group for the last, it's more than 15 years now, even before at the same time I think I began approximately about 15 years, is the problem of Indo-European language origins. So Indo-European is a language we're speaking now, English. Almost all European languages are Indo-European languages, and it was noticed more than 200 years ago by several people, including William Jones in Calcutta, who knew Greek and Latin from his schoolboy days and began learning Sanskrit, that these languages have many shared features, including words and grammars, and that they might stem or must stem from some common source which perhaps no longer exists, and the question was where that might have been. So until 2015, the large-scale view amongst most archaeologists was that it was farming that spread these languages. We know of an economic revolution that happened in Europe and the Middle East and South Asia beginning 11 or 12,000 years ago in the Fertile Crescent. And after 8,500 years ago, that explodes into Europe, and it's transformative economically. And the settlement patterns change, so that would be a natural way that people could have moved into Europe carrying new languages. And there are also similar impacts to the East. So Colin Renfrew, in this book, argued that this is the spread of Indo-European languages, probably out of Anatolia, what's today Turkey. So beginning in 2015, that picture completely fell apart as a result of ancient DNA. So here is a picture from a series of papers that we wrote that shows how people today relate to each other. So this is a scatter plot with an X and a Y axis I'm not showing you, and each dot is a person living today labeled by where on the map of Europe and the Near East people come from. And the data is whole genome data, so it's about 600,000 positions in the genome where people differ from each other, and we multiply the table of 1,000 people by each other to see how closely related each person is to every other. And then we display the variation with principal component analysis, which most efficiently separates the people based on how much variation is explained in that matrix of how closely related people are to each other. So what you see is this amazing pattern where people splay out without using any labels in the analysis in two gradients with relatively few people between them. There's a European gradient on one side, and there's a Near Eastern gradient on the other side. And in between are very few individuals who are plausibly the result of more recent contacts between Europe and the Near East. So this is mostly people who are Greek and people who are culturally Jewish. On the top, you see people who are non-Mediterranean, and on the bottom, you see people who are Mediterranean. Now I'm going to gray out the points, and I'm going to show you where the ancient people fall on the same plot, just learned from the modern samples. The hunter-gatherers of Europe, the ones who lived before the farmers arrived, are beyond Europe in the direction of European differentiation from the Near East. The only possible explanation for that mathematically is if people in Europe today are mixtures of people like this and people more Middle Eastern. So there was some mixture occurred. We don't see people who are like this anymore because their ancestry has been mixed into people like Europeans. Then the first farmers arrive and they pile up at the bottom of European gradient on the Mediterranean side, but you don't see people like most Europeans today. For example, English people are over here. And then about 5,000 years ago, on the steppes north of the Black and Caspian Sea, these mobile steppe pastoralists appear here. And archaeologically, they then explode. They explode to Hungary in the west, and they explode to Mongolia in the east. And after 5,000 years ago, boom, you see Europe form. There's a massive mixture of people like this and people like this and a little bit like this to form the gradient that we see today. And ever after, it looks like that. And what this is the spread of these mobile steppe pastoralists into Central and Western Europe, mixing with the farmers to produce the gradient that is there today. And the argument that these steppe pastoralists had not made an impact was that once farming was established, they could not have made an impact. How could thinly spread herders make an impact on a densely farmed area? Similar to how could the British make an impact on India, which was a dense place? They didn't make a demographic impact, but these people did. The data show it. And it's a large impact. It's about 3/4 in Germany, it's 90% in Britain, and so on. Here's a few examples. Yamnaya people spreading Indo-European languages west. If you look in Britain, there's no Eastern ancestry from the steppe until about 4,500 years ago. The last big stones at Stonehenge go up, built by descendants of the first farmers who arrived in Britain about 6,000 years ago, and then boom, it's a 90% population replacement. In Iberia, in the same time transect, here's the first farmers 6,000 years ago. They arrived there earlier, but I'm starting the time transect at the same time. 4,500 years ago, you still don't see people from the east, and then suddenly you see them, but here there's a time when they're overlapping, and then they collapse into each other 4,000 years ago. The dots are colored by the Y chromosome of the people and whether it's a steppe eastern chromosome. And what you see in Iberia is that even though it's a lower mixture proportion, only 40% migration from the east, there's no local Y chromosomes left. It's all turned red. And what that means is that the local male population completely failed to transmit its Y chromosomes to the subsequent population. And so how that happened, we don't know, but several thousand years later, the descendants of these Iberians came to the Americas and exactly this happened. Today, people in Colombia have almost no local Y chromosomes. They're almost all European, and they have almost no European mitochondrial DNAs. They're almost all Native American. And that's the result of exploitation and social inequality, and perhaps that's what occurred here. These Yamnaya people, these steppe pastoralists, you can see their DNA spreading throughout. We can see and track it all across Europe and into South Asia in time and place, often matching the reconstructed branches of these languages as learned from linguistics. It's very hard to believe that the spread of Yamnaya is not what spread these languages that are spoken today. But it's super complicated. We don't always see spreads of genes matching culture. Here's a paper we just published this year on the Phoenicians. So Phoenicians are these traders of the Iron Age who lived in what's now Lebanon, and they then were succeeded by the Carthaginians, which are Phoenician colonies in different parts of the Eastern Mediterranean, Western and Central Mediterranean, marked here, especially in North Africa, but also in Sardinia and Iberia and Sicily. And a question had been, like, how are they related to each other? Certainly they spoke the same language, they shared the same religion, can we get DNA from them and see? And so we got DNA from a couple of hundred people and looked, and they're completely unrelated. So the Phoenicians cluster here on top of earlier populations from the Middle East. And the yellow people are the Punic people, the Carthaginians. They're just almost completely non-overlapping. There's almost zero contribution. Greek people are over here. North Africans are over here. Carthage is a North African part of the Mediterranean, and people in Punic sites across the central and western Mediterranean are a mixture of these two groups with mostly Greek-like, Sicilian-like ancestry and some North African-type ancestry. And it's not more in North Africa than it is in Sicily or Iberia. It's like a cosmopolitan population that's swirling around this part of the world. The Carthaginians were largely genetically derived, it seems, from the same source as their Greek commercial rivals, which is kind of amazing. And it forces even authors of our own paper to change their assumption. There was this long-standing debate is, should you even use this word the Romans used, Punic, to refer to the Carthaginians of the central and western Mediterranean? They're really Phoenicians, but genetically they were completely different by the time we were able to sample them. Another example from this disruption that you get from ancient DNA is about what white people are. So greater than 8,000 years ago, in the place where white people live today, we actually can sample DNA, and there's 4 groups, 2 hunter-gatherer groups, 2 farmer groups, that we can get data from, and they're each as different from each other as East Asians today. So how did that happen, and how is that related to what you see today? Well, nobody disappeared, they just mixed with each other. That's people living after 4,000 years, and today the low level of differentiation is just due to mixture of these 4 groups. Here's another example. There's so many examples, but the Southwest Pacific, so Vanuatu, there's a whole series of papers we've been involved in where the first people of the Southwest Pacific are almost entirely East Asian in ancestry, so no Papuan ancestry, but today people are about 90% Papuan ancestry. That's because the first people are almost entirely East Asian. They didn't pick up almost any Papuan ancestry on the way to getting to the Pacific in these outrigger canoes and navigating long distances, even though they passed Papuans. But within 500 years, there's a flood of Papuan ancestry, primarily mediated by male Papuans, mixing with the local population, and it results in this rapid and long-standing established change. So the big perspective change from ancient DNA study is that people living today are almost never the descendants of the people in the same place thousands of years before. There are exceptions, but it's, the rule is that. Everyone's mixed, and no one is or ever really could be pure in any sense. When we collect DNA from previously unsampled times and places, the Denisovans and many other places, even within the last 10,000 years, major human lineages are found that simply don't exist anymore in unmixed form, or sometimes haven't left very many descendants, but they were widespread at the time. And large-scale human movements have occurred at multiple timescales, often disruptive to the populations that experience them, and these patterns are not possible to predict and anticipate without direct data. So 3. Before ancient DNA, we had a fundamentally wrong understanding that natural selection has been quiescent in the last 10,000 years. So here's a paper from 2011 that says classic selective sweeps were rare in recent human evolution. So this is a genetic variant that arises due to a new mutation and is advantageous and goes up to 100% frequency because it's advantageous. And there's several lines of evidence for this. The one in this paper is very interesting. I'm not going to explain it. Another is that if you compare Europeans and East Asians, there's almost no genetic differences that are completely different in frequency across them. If there had been strong natural selection, you would expect a lot, but there are not. So for example, just a year ago, Sasha Gusev wrote you should read his blog because it's extremely interesting and thoughtful and valuable. This blog, which is, where are the recent selective sweeps? How do we make sense of the surprising few loci under natural selection? So ancient DNA studies, a lot of the interest in ancient DNA was to discover biology, not to discover history. The biggest interest was what makes humans different from Neanderthals biologically? Can we see what's become differential from other groups? But the first discoveries were really about population mixture and migrations. And the main reason for that, perhaps, is just that you can learn so much from a single genome, because a single genome is not just one person, it's a multitude of people. It's your parents and your grandparents and your 8 great-grandparents and your 16 great-great-grandparents. Each bit of DNA reflects one of those other people, and so you get tens of thousands of independent pieces of information showing how people are related to each other. You get an exquisite information about relatedness of people. And so one single genome, you can position each person exquisitely relative to others. But to study frequency changes of genetic variants over time, to understand biology, you need a large sample size. And we didn't have that at the beginning. So we started to look. So we published a paper in 2015, and we looked for places that had changed unusually over the last 8,000 years. We found 12. A study just last year found 21 in a bigger sample size with higher quality data, but it felt like it was maxing out. So we worked in the last 6 years to try to improve this. So we developed several innovations to increase power and to reduce false signals. One was to develop a new statistical method that didn't involve modeling of demographic history, but just used the data to see how everybody was related to everybody else and control for all these mixtures and migrations. Another was a very much increased sample size. This study is releasing data from more than 10,000 people in a single study. And to greatly work on data quality through all sorts of data cleaning. When you now apply this to the data, we found almost 500 places that are genome scale significant. So this was extremely weird and surprising to us in light of the previous literature, and we spent years trying to make this go away. But it kept getting stronger. It sort of felt a little bit like what we had done with the Neanderthal work and the evidence of mixture. So how do we reconcile this observation of this vast load of genome scale significant findings, about 500 of them, and we think that actually there's at least 5,000, but many of them are sub-significant, with the lack of evidence for selective sweeps? Well, one answer is we're definitely in a period, at least in Europe, of intensified evolution. So what we can see is that there is enrichment for evolution for genetic variants that we know from genome-wide association analysis affect immune traits and metabolic traits beyond background, and that there's an acceleration in that enrichment statistically after 5,000 years ago. So we're in a period of accelerating biological change in this period, provably based on the ancient DNA data. And the second answer is that their selection is fluctuating over time. So if you plot the true ages of the mutations that we find under selection here against their expected age if they had been under positive natural selection the whole time, you would expect it to be on an x equals y line, and about a third of them are, but about 2/3 of them are not. And in fact, the red ones have reversed direction, it's the ancient allele that's being positively selected. So what's happening is these are genetic variants that were existing in the population before that have come under a different type of selection and reversed. And so the reason they haven't become 100% different in frequency between Europeans and East Asians is that there are variants that go up, and then they go down, and they go up, and they go down in different environments. Here's two examples for celiac disease. This is related to gluten insensitivity, which both shot up in the last 4,000 years. These are risk factors for celiac disease. They probably do something valuable in addition to causing risk for celiac disease. Here's a major risk for tuberculosis, for clinically severe tuberculosis. It's now at a few percent frequency, but 3,000 years ago it was 9%, and it rocketed downward under the pressure of selection. But before that, it rocketed upward. You can see the change, and both of these are significant. And I don't know, maybe tuberculosis becomes endemic, and that's why it decreases in frequency. You can also look at combinations of genetic variants that shape complex traits. So for studies of complex traits like diabetes, you can look at tens of thousands of people with type 2 diabetes and tens of thousands of people without and look to see which genetic variants affect this trait. And you could do a linear combination of the variants that predict risk for type 2 diabetes to make an even better predictor, a sum of about 1,000 variants, for example. And you can watch how that changes over time, and you can ask, does that combination of variants, that's a good predictor for type 2 diabetes risk, change over time more than expected by chance? And what we see is that that's pushed down by about a standard deviation on the scale of current variation over about 10,000 years, clearly under the pressure of selection, as well as type 2 diabetes-related traits. You also see this for some psychotic-related traits like schizophrenia, where there's clear selection against what today manifests as schizophrenia. It does not mean schizophrenia was more common in the past. It could be that environments today are more likely to cause schizophrenia than environments in the past, but it does mean that people with the genetic inheritance of people from 10,000 years ago put in today's environment would be more likely to get this. And there's also traits like that are years of schooling, which, believe it or not, have strong genetic predictability that have increased over time, in this case, by about 0.6 standard deviation. We tried to make results like these go away, and we wondered if they were a statistical artifact, perhaps of inadequate correction for migrations and mixtures in our study. But they replicate, for example, the years of schooling one, when we measure the effects of the variance on years of schooling in East Asians and correlate them to the trajectory in West Eurasians, two groups that have almost completely uncorrelated population structure. It's very hard to see that that could be a result by chance, and they replicate when we measure the effects of these in families where it's very hard or almost impossible to see how that could be the result of population structure. So the summary of the talk is that this is 3 examples of how ancient DNA has really forced our hand, challenging understanding of the world in deep ways. Before ancient DNA, we had a fundamentally wrong understanding of how modern humans relate to archaic humans. Before ancient DNA, we had a fundamentally wrong understanding of how human populations migrate and mix with each other. And before ancient DNA, we had a fundamentally wrong understanding that natural selection is being quiescent in the last 10,000 years. Thanks. This is photographs of people currently in our research group here. And there are many past lab members who contributed to this work, and many collaborators and funders. And now I'd like to invite Carl Zimmer to join me for a conversation.
Thank you, David. So, as was mentioned earlier, we will be reading some of your questions. You put your questions in, whether you're watching online or here in the room, using Slido, and I will see them here. So I will just start things off by asking just a few questions of my own. You can sort of be absorbing the 60,000 years of history that you just learned about while I'm asking. So I'm sort of curious. I don't think I've ever asked you. Do you know what percentage Neanderthal you are?
I've never sequenced my own genome, and I've never sent my DNA to any direct-to-consumer ancestry testing company. I don't know if it's stubbornness at this point, but I've never done it.
I'm curious, in the audience, maybe would anybody be willing to raise their hand? Do you know, how many people know how Neanderthal they are? Now look at that. Isn't that interesting? Like, because of the research that you and Svante Pabo and others did, like, individual people have been able to see an estimate of how much Neanderthal DNA they have in their genome. What's that like for you to sort of see that kind of impact?
That's an interesting question. I don't know, it just seems very important, this stuff. It just seems fundamentally important, so I don't really think about it that way. It just seems like an important set of facts about the world that we're related to archaic humans in these ways. And so I don't think about that. That's not a very helpful answer.
Well, no, it is what it is. I mean,
it's just so fundamental to who we are that we're related to archaic humans. So,
but I would say that. But I would say, what I've found sometimes is that people make the mistake of thinking I'm a scientist. But since I write about this stuff, they'll say like, well, I'm 2% or I'm 1%, and they think that number has a lot of meaning for them in a very deep, intimate way, because it's their own DNA. Does it make a difference?
No, it doesn't really make a difference. And I actually think a lot of those numbers are exaggerated. So as far as I can tell, I don't know what the current estimates are. They may just be counting informative positions at some point, but the actual estimates we obtain have statistical errors about 0.5% on a per individual basis. And the actual theoretical variation is about 0.1 or 0.05%. So I think people are just quoting noise mostly if they're non-Africans. If they're African-American, it will be really reduced because they'll have mostly West African ancestry, for example. But I think people are really, to a large extent, just interested in, if they say, oh, I have 3% Neanderthal ancestry, it's probably just a 2 standard deviation variation just from the measurement, not from the actual real inheritance.
At the same time, though, like we have different, my Neanderthal DNA is not identical to yours. We may have different Neanderthal alleles. Does that, based on people looking at the surviving Neanderthal alleles in the human population, does it seem like it can make a difference to people, like, comparatively, like, experiences with health and so on?
Neanderthal, you have about 2% of your DNA is from Neanderthals. 2%'s a lot. The Neanderthal DNA is actually especially different, so it's loaded with variation compared to random variation in the genome. It's also been selected against, so that depletes its biological significance on average. So it's not clear whether on average that 2% is more or less important than average. There's, I think if I remember right, about 2,000 segments of Neanderthal DNA estimated in, if I remember right, I may get this number off, in each non-African person. And so the error around that number is very, very small. So almost everybody will have just within, 3% of that number, just by chance.
Now, looking at that sort of big picture of human evolution, the tree that you showed, with Neanderthals and Denisovans and perhaps other branches, you were arguing that we need to think harder and think about what's missing there. And I guess one question that comes up when we look at a tree like that is that there's so much diversity in our humans and their relatives. 100,000 years ago, 50,000 years ago, And as you mentioned, there's not that much now. So do you feel like studying ancient DNA can give us any clues as to why so much of that diversity went away, why there are not clear-cut populations of Neanderthals or Denisovans or other kinds of humans around anymore?
I think it gives us a lot of clues. I mean, I don't know if we know what happened when Neanderthals and modern humans met and mixed. The Neanderthals as groups disappear, and they get mixed into modern humans in Eurasia. Whether that was violent or whether that was just environmental degradation, we don't know. But I think documenting these events as they occur through ancient DNA allows us to obtain a much richer picture of what happened. In principle, one could analyze the genomic distribution of segments of Neanderthal DNA in modern humans to learn something about the nature of the event, kind of like we study in Iberia, and you see that there's very little local Y chromosome contribution from the local population. Maybe you could hope to do something like that, but there's so much natural selection that's shaped the bits of DNA from Neanderthals after it that it distorts the picture and it makes it hard to read that off. One of the things I wanted to say in response to your sort of comment about that very complicated tree is that that tree is the result of an accretion of findings. So one finding after the other established one of those lineages or mixture event, and they kind of got grafted onto each other. And it sometimes reminds me a little bit of the sort of old models in like 2,000 years ago, like these Ptolemean epicycles, adding things together to make the solar system seem to work still. And one wonders whether there's a reconfiguration that might make things work differently. Just another one, when the Denisovan genome was first obtained in Svante's group, the mitochondrial DNA is like 800,000 years diverged, or 700,000 years, or 900,000 years diverged, and it seemed to be an outgroup, a more distant relative of both modern humans and Neanderthals. Modern human and Neanderthals were sisters, and Denisovans was much more distant. But then the whole genome was sequenced, and it was very clear that Neanderthals and Denisovans were sisters. And I think we were in an age at the time of superiority of the genome. So if you have the whole genome, it tells you they're most closely related, then that's the main story. But actually, there's a diversity of stories. Each part of the genome tells its own story, and they're all valid stories. And if the Y chromosome and the mitochondrial DNA tell you a profoundly different story, why should we ignore them? They may be numerically less important, but that might be because of some other biological or demographic process that if we think hard enough, we can figure out. So it's not really at all clear to me what the right solution is. It's just that the current picture feels so wrong to me.
Looking in fossils and maybe finding some really interesting ancient DNA might.
That would be, that is often how it gets solved when a unique specimen is obtained and resolves it. There's a very exciting specimen that's been sequenced in various forms in Leipzig and Svante's group from Spain, Cima de los Huesos, and I didn't talk about it very much, but it's surprising. It constrains the story and makes it difficult to come up with solutions that are alternative.
So let's leap forward in time. You spoke about the Yamnaya in terms of like understanding sort of how European history was shaped by these people coming out of Central Asia. And and the diagram you're showing is quite dramatic of just these people that perhaps most of us have never heard of before this research suddenly being everywhere. So can you connect what you're seeing in the DNA with some sort of hypothesis for how it is that some group out in the steppes suddenly is everywhere?
Yeah, that's a great question. I think archaeologists thinking about this there is burial traditions that are associated with the Yamnaya that spread very dramatically. And archaeologists, even the strongest proponents of the Yamnaya being involved in spreading into European languages, really couldn't bring themselves to argue that this was really a truly large number of people. It seemed unbelievable. So they would argue, well, this is maybe an elite cultural tradition that gets learned and adopted by neighbors and sweeps through bringing people's languages. It's like the same reason, for example, Hungarian maybe gets adopted even though there's not a large-scale demographic replacement by people from the East. Maybe that's what's going on. Nobody dared to say there were a large number of people, but the DNA scream at you, there's a huge number of people associated with this. There's populations collapsing and being displaced or admixed with these groups that are expanding. People make arguments about maybe very early use of the horse by the Yamnaya might have given them an advantage. There's arguments about whether they used the horse uniquely or in a truly domesticated ways. Their use of wagons, the use of this steppe as a food source. So people make these arguments, but the actual observation that they made that expansion is just an unbelievable thing.
So the kind of research that you do depends on, the data that you can get access to. And a lot of data, for all sorts of historical reasons, has been coming out of Europe, either from living Europeans and things like the UK Biobank or other kind of efforts, or those museum collections of European specimens. We're talking about, over the past 10,000 years. I'm wondering to what extent when you see that process of migration and populations overturning and so on in Europe, how does that compare, insofar as we understand it, to what's happening in parts of Asia, like India, or in Africa, in other parts of the world?
Right. So we are 5 to 7 years behind, depending on where we are, in terms of what's happening with European ancient DNA in some of the places you mentioned. But it's exponentially growing also in sub-Saharan Africa with working data in places that are very hot and you might not expect preservation to exist. And in the places in sub-Saharan Africa, we've gotten data from many of the same types of patterns that are observed. Example 1, you get DNA from Malawi in South Central Africa 3,000 years ago to 8,000 years ago. There's a population there that's separated for 150,000 years from everybody living today that barely contributed to anybody today. It's established there for a long period of time. And then there's an expansion of people, ultimately probably from areas around Cameroon, associated with Bantu languages, that's almost a complete displacement of this group. You look in Cameroon itself, you think this should be where these people come from. 3,000 to 8,000 years ago in the rock shelter in the grasslands area of Cameroon, it's supposed to be the homeland of the Bantu expansion. They're completely different from the Bantu. So everywhere you look, where you get data from places that have not had data from before, people are different in the past from how they are today, either completely different or mixed with the present-day populations. So certainly in different parts of sub-Saharan Africa, we're seeing these patterns similarly to Europe and the Middle East. And East Asia, the same. Not very good data from South Asia, but it seems likely for multiple reasons, and the little data we do have, that it's the same. Patterns are qualitatively somewhat different in the islands of the Pacific, even with a short time, often the same, as I mentioned. In the Americas, it may be qualitatively different with less frequent turnover.
What do you think are when populations are getting replaced? I mean, what do you think are sort of the plausible, possible explanations? Warfare, sort of out competition. Just that some populations disappear due to disease and other populations just move in to where places where the other people were? Like what seems to fit with what the DNA is saying?
Well, this is speculative, of course, but certainly the case in Iberia I told you about can't have been happy occasion for the men involved, the local men involved. They did not succeed in transmitting their Y chromosomes to subsequent generations, whether that's due to the local women preferring foreign men, or whether that's due to the local men being, dying in some way, we don't know. But the sex-biased admixture is sort of a canary in coal mine sign of inequality or unequal or interactions amongst communities. We could also think, and actually it's a useful thing to think about the Neanderthal case in Europe. Neanderthals disappear pretty rapidly over a few thousand-year period across Europe, and actually it's a wave of loss. So in certain parts it's earlier, in other parts it's later, but in any one part it might be relatively quickly. And so you might think a simple story like is dramatized in some of the novels about this, like that the modern humans are out-competing the Neanderthals because they're more violent, their culture is more aggressive, or maybe they have some technology, even though Neanderthals are very sophisticated, and who knows. But actually, what's probably happening is like lots of extinctions locally of populations for various reasons. And most of the modern human early groups in Europe also disappear. So the individuals from Bachokiro, the individuals from Oase I talked about, and other individuals, they're from lineages that didn't contribute substantially to later Europeans. And there's 39,000 years ago, there's a climatic fluctuation that results in almost everybody who was living there before in Europe, including the Neanderthals, but also including all the modern humans, seeming to disappear. And they're replaced by more humans from probably the Near East or maybe one isolated group in Europe. So it's not clear that it's a systematic thing, but rather many, many groups going one way or the other.
Just my last question before I go to the questions from the audience. So you describe a sort of a myth of purity. I mean, do you think that that people today, do you think that that is a widespread myth? Do you feel like people have this image of themselves and their heritage as being quote unquote pure? And if so, how do you think that people should incorporate the findings of you and your colleagues to really think about their heritage?
I think many people have an idea of purity of their lineage going back a long time, and perhaps giving them rights to live in a place or so on and so forth. You see this in India, for example. You see this in parts of Europe. You see this in the Middle East, and so on. And the truth is that people are layers and layers of mixture and migration, and I think that the ancient DNA shows this as clearly as it does that all non-Africans have Neanderthal ancestry. I think it's one of the things that we can do in this field is make it clear to people that this is true. You can't assert anymore that people have been living in pure form in Europe for 15,000 years, 20,000 years, 40,000 years, because it just didn't happen. We have data from different times and places, and the truth is just layers and layers of people moving and mixing from diverse places around the world, not the places that people thought they were coming from. People's stories about their history are almost always wrong, and I don't think that that's a bad thing. I think that's a good thing and a humble thing to be made aware of.
But it can be hard to give up those stories or to modify those stories in the face of information.
I'm not sure I understand.
Well, I'm just making an observation that these stories about people's past, even if they are incorrect, it seems like it's difficult for people to actually modify their stories to take into account what science might be telling them.
I think what science can do is to establish things as scientifically accurate, and that's a very powerful thing in this world. So you can be in the realm of telling stories about the past that you don't invoke science, but people sometimes say, well, science has shown, or science probably shows this or that story about the past, and now that we can measure it, and we know, A, that people's stories are almost never what they think they are, they almost certainly don't match these accounts. I think it constrains this, it marginalizes this discussion from the ability to claim scientific imprimatur.
Okay, so let me get to some questions. So at the end of your talk, you were talking about strong natural selection in the past few thousand years. And you're seeing connections with diseases. And so we'll take the celiac example. So someone who was asking, is there an understood value or benefit associated with celiac disease to account for the change in the penetration of the phenotype? So why is it that you see a gene that's associated with celiac disease increasing? Why would that happen?
Don't know. So
All right,
this is
there you
what
go.
this is what the science, this is what the data tells you. There's a change over time in very strongly under clearly incredibly strong selection pressure for a major risk factor for celiac disease, not just one but multiple, over the same time period. This is like gluten insensitivity. If you made up a story in a wheat-using world, maybe you would expect the opposite. So maybe it's doing something else, protecting against some infectious disease that's become prevalent in the area. I think that there's very few examples of natural selection where we actually understand the biological mechanism. So we can take this rapid increase in celiac disease and dissect it molecularly, as, for example, the EDAR variant in East Asia has been dissected, and the lactase persistence. There's a small number of cases where people have wrestled these to the ground and know a lot more. And I think this provides grist for that mill.
So there could be something that comes with this mutation, which is a benefit, could have been a benefit, in some early environment, even if, maybe later on it leaves people vulnerable to some type of disease like celiac, or there have been studies on multiple sclerosis and so on. So
we have 3 examples of this, that are very clear. So one was this TIC2 variant for tuberculosis. We see a reversal. So presumably this is an immunologically active variant that did something useful for people and was under intense positive selection. Then it was exposed to something else, plausibly TB, which is, some people think, is the biggest killer, infectious disease killer, and it caused a reversal because it's a risk factor for that disease. There's a balance between different immunological pressures. Another one is hemochromatosis, a major risk factor, hemochromatosis. It reverses around the same time, 3,000 years ago, positive selection before, negative selection afterward. In the multiple sclerosis case you talked about, the previous study argued it was under positive selection, but with this higher quality data, you can detect a reversal, actually, again, 2,000 or 3,000 years ago, especially in Northern Europe. So it's actually not advantageous, and it goes in the other direction.
Wild. Okay, here's a question that I have asked you this question more than once over the years. I've asked Svante Pääbo this question too. I have not gotten an answer from either of you. Understandably, I totally understand, but since someone has asked it, I will ask it again. Are modern humans and Neanderthals considered to be one species or different species?
It's not my department. Sorry, the answer...
Whose department is it?
No, so the answer is, what is a species? So the biological species concept defines a species as groups that don't in practice mix when they encounter each other. As the data shows, every time Neanderthals and modern humans encountered each other, they mixed whenever they could. So this was not a group with a barrier to gene flow in it, and in fact, they left a major legacy in people living today. So if your definition is the standard biological species concept of reproductive barriers, then they are not different species, neither are Denisovans. But they did have evidence of hybrid incompatibilities, as I described to you. I remember a discussion in our collaboration, if I'm allowed to describe it a little bit, when we were publishing the Denisovan paper. There was a constituency which wanted to call it a different species. There's some kind of excitement amongst people who study the past in naming a species. And there was a constituency that amongst the co-authors who wanted to name their own species. And it was an extremely interesting email exchange. And I remember the email that Svante wrote about this, which really argued that we're going to use a common name, a group name like Neanderthals, rather than a species name, which makes a judgment about reproductive barriers or so on, because we're finding that there's interbreeding, and it almost doesn't really matter anymore to define them as a species or not in this context. I don't think we're dodging it. I think we're just saying it's very confusing, and in fact, many of the common definitions of species don't apply because humans really mix with everything they can mix with if they can.
Our defining feature, I guess.
Did I answer your question?
Kind of. So here's a question. You did not mention ancient hominids from Southeast Asia who were diminutive. This would be, I'm going to assume, there are these fascinating fossils. One's called Homo floresiensis, like about that high. lived maybe, I'm trying to remember, maybe 50,000 years ago or less. And there are other diminutive hominids from the area too. So, this is in the same time window as Neanderthals and Denisovans. These are really fascinating hominids, not just in Southeast Asia, but there's Homo naledi in Africa and so on. So, no DNA from them yet?
No DNA that I'm aware of. It would be amazing to get DNA from them, and I'm not aware of any DNA. But when I said there's 5 groups 70,000 years ago, at least as different to each other, much more different to each other than any groups of people today, I was counting Floresiensis without DNA, and then I was counting at least 2 groups of Denisovans, the ones from Siberia and the ones that intermixed with the ancestors of Melanesians, and then Neanderthals and anatomically modern humans.
Yeah. So when scientists like yourself say, we've looked for ancient DNA, we haven't found it, should we take from that, like, there's not any DNA to find in these remains, or is it possible that there's still an opportunity for some breakthrough method that will be able to retrieve DNA from fossils that once seemed to have no DNA in them?
I think there's lots of opportunity to get DNA from these fossils, even in tropical areas. I think the best methods should be applied. They should be applied repeatedly to lots of different specimens, to the ones most likely to yield good DNA. It's such an amazing yield of information to get a genome, or even to get fragmentary parts of a genome, that in my mind, it's definitely worth doing. So even though we haven't seen any data reported yet from some of these unique and special and important specimens, I think there's no reason to think that it might not work with modern methods.
Let's see. I'm just trying to. So, so if we had, this is an interesting question. Is there any tension between physical anthropologists and archaeologists versus the work that you and your colleagues do, which overturn long-held assumptions? So you're coming from this sort of genetics background. You collaborate a lot with archaeologists and anthropologists, but you do come from these different disciplines. And sometimes you're saying, look, my data tells me something that says this idea that you've had for a long time is wrong. How do those conversations go?
Yeah, it's a great question, and that's the theme of this talk. So this is really interesting new information, and it's wading into an area like, archaeology of the steppe, or Indo-European languages, of which there's been no data before. You simply couldn't look before at how these people are related to each other, and the data falsify arguments that people have been making for decades in PhD theses. You just can't sustain these arguments even if they were reasonable before. And so it's been very interesting to be part of that. I'm not learned and schooled in, Anatolian linguistics or in, Punic archaeology or something like that, but we bring to bear unique data that is relevant to these topics. And so the question is, how do you have that conversation? So often the conversation starts in a very crude way. We get some specimens, we sequence them, and we say something that sounds very off base to specialists in the field, something they would never say themselves, having gone to the graduate school. So hopefully there is a process by which we have friendly archaeologists and anthropologists we're collaborating with who will yell at us quietly and will help us to form a final manuscript that can communicate to these other communities. But it doesn't mean that we should not say strong things. So one of the dangers that I've been seeing in the field of ancient DNA is a kind of desire to just say in agreement with this field, in agreement with what's been seen before. But actually one of the powerful things this technology can sometimes bring is in disagreement with this prevalent view. We can make progress by identifying a set of theories previously plausible that are not plausible or very much less plausible. So that's happened with physical anthropology, especially in the context of the Neanderthals, for example, where there were really interesting tensions. And these are the specimens that they've curated that we're studying. This has happened with archaeology, and I think there's an ever richer dialogue that's really very positive. Archaeologists are super thirsty for information about the past, and it's beginning or actively happening with historians, and these disciplines are becoming further and further from ancient DNA. History is really in the humanities in many ways, and so we really need to bridge that sort of disciplinary gap. You know, some people who don't even believe in objectivity, for example, or some people who, you know, question various assumptions or who have profoundly different ways of writing with great respect for these other disciplines. So it's actually fun. You know, it's like a privilege to talk to these people in these different communities, and it's often kind of tense and interesting ways that one can learn from.
Great. All right, well, thank you, David, and thank you all for coming. I'd like to ask Tomiko to close out the event.
Well, I want to thank David for that fascinating lecture. That was wonderful. And thank David and Carl for the thought-provoking conversation. And thank all of you for joining us and for your questions. I hope you'll join us again soon. You can find more information about upcoming events on our website, and I hope you have a wonderful evening. Thank you.